Okay, so this is chapter 13. This is part A of the peripheral nervous system, so the PNS. And so what we've been discussing in chapter 11 and 12 was the CNS, central nervous system, the vis-a-vis the brain and spinal cord. And so now our focus is going to be the peripheral nervous system. And so we're going to look at really what that means, you know. So again, the PNS links inside, outside body, right? Basically what we're seeing here. It links the CNS, in fact, through the PNS. So again, your PNS contains both sensory, right, and motor neurons. And those, in fact, sensory and motor neurons will receive information. Central nervous system processes that information, sends out responses to it, and then the motor neurons send that, carry that signal down to effectors, right? In fact. So this is really what we're seeing. This is what we mean by linking, to and from the outside world, right? And so we're going to look at part one. We'll talk about the sensory receptors, right? And the different kinds we have, generally speaking, in the body. Then we'll take a look at how nerves, how we structure nerves, how we repair nerves, in fact, because the PNS can be repaired, and that's limited. Most other neurons in the whole nervous system cannot be repaired, but the PNS has a limited repair feature, right? We won't really take a look at motor nerve endings and motor activity per se, and we won't really take a look at reflex activities, but we will talk about the first mainly, okay? So again, here's our breakdown, right? Here's the PNS, and it's, two divisions, sensory, motor, and then the motor having somatic, autonomic, sympathetic, parasympathetic, and that breakdown also, right? Okay, so again, this is part one. We'll talk about the receptors per se, and then the process of. So again, sensory receptors, right, respond to changes in the environment. They're going to pick up stimuli or stimulus, right? And what we mean by that is actually we activate the neuron. We see a greater potential become a nerve impulse, right? So we see a localized change in ions becoming a larger change in the neuron or nerves, sending that long signal down and then up to the CNS, right? So what does this give us? Sensation, right? The definition of sensation is the awareness of the stimuli. Okay, so if you know consciously the stimulation or the stimuli, then this is what we mean by sensation. We also then convert that sensation to perception, right? Perception. And that's the interpretation of the meaning of the stimulus. So you have sensation and then you have perception, right? Sensation is done by the sensory neuron. Perception is done by the central nervous system. So this is PNS versus, in fact, CNS, right? And again, just to kind of throw the theme again, where sensory integration and then motor responses occur as part of the nervous system, right? So we're going to look at receptors. We're going to look at types of receptors. And then you always have to keep in mind that the types of receptors are classified based on the type of stimulation, where it's located in the body, and the structure of the stimulus, right? So we're on the sensory side. Okay, so we've really discussed these before, but they're very easy to understand because they're actually two words in one, right? There's your mechanoreceptors, thermoreceptors, photoreceptors, chemoreceptors, right? Mechanoreceptors, again, think of something physical, right? Touching, pressure, vibration, stretching. Mechanoreceptors pick that up. Changes in temperature, thermoreceptors pick that up. Changes in light, photoreceptors pick that up. And changes in chemicals, chemoreceptors pick those up. So again, this is the type of stimulus, right? We can classify them based on this, right? Nociceptors are a special type of receptor because it picks up pain, right? So what do we mean by pain? This can be temperature changes that are extreme, pressure that's extreme, chemicals that cause inflammation can be picked up by the nociceptors and interpreted as pain, right? So these are pain receptors. Okay. These talk about location, right? Okay. External receptors, keyword is outside the body, right? So these receptors are responding to stimulus outside the body, which can be nociceptors, mechanoreceptors, chemoreceptors, etc., right? So again, this is based on the location, right? Most of your special senses are going to be external receptors, right? And what do we mean by that, right? Vision, hearing, taste, smell, right? Those are your special senses, which then have external receptors responding to the outside of the body, right? Interoreceptors or visoreceptors, think of the word visceral, right? Because that's why we use the word visceroreceptors. This is stimulus, then on the opposite, inside the body, right? Internal viscera in the blood vessels is where we have internal receptors, right? Again, they can perceive or they can interpret the same type of stimulus, except location is going to vary, right? So chemicals, pressure changes or mechanoreceptors, temperature changes or thermoreceptors. So all these internal receptors can also be those types of receptors. Sometimes we can perceive them as painful or discomforting, in fact, irritating, but usually these are going to be unconscious, right? We don't perceive them consciously. So they're going to work, in fact, on a subconscious or unconscious level, which means we don't feel them receive the stimuli. We interpret that information in the brain, but the brain doesn't interpret it consciously and say, hey, this is a temperature change, per se, right? Proprioceptors, which we've talked about, which you find in skeletal muscle, all your connective tissue around muscle and bone. And those really identify movement of those skeletal muscles, bones, joints, tendon, connective tissue in space, right? So again, these are the ones that go off when we move our hands or we move our legs or we move our torso or we bend down, bend up. These are the receptors going off, telling the brain where those body parts basically are at in space and time. Okay, so as far as the structure now we're looking at types of receptors based on structure. They're usually going to belong to one of these two categories, right? The receptors for general senses and the receptors for special senses. So the key word is general sense versus special sense, right? Okay. So the general senses are going to be your sensory neurons, right? General sensory information. Okay. pretty much found throughout the body. Monitoring all of this, monitoring all of this information, generally speaking, right? And the special senses, again, you see those here, right? Which is the list I put down with the exception of equilibrium. I did not put that one down. But that's your position of your head in space. And then we have special sense organs for those special senses, one of which we're going to dissect this afternoon, the eye. So we'll see a special sense there. But nonetheless, those are your special senses, right? Okay, so general senses, right? Touch, pressure, those are all your tactile sensations. Temperature, pain, muscle change as far as contraction or stretching. So the interesting thing about general senses is they can actually respond to multiple stimuli. So they have the capability of receiving not only, for example, tactile sensation, but temperature, pain, and et cetera, et cetera. So they have the capability of multifunction, right? So in the general senses, we're going to either have the receptor, what we call being a free nerve ending, or non-encapsulated versus encapsulated nerve ending. So just this depends on what its function is going to be, if it in fact has a capsule on it or not a capsule. So here are the free nerve endings, non-encapsulated. You find that in the epithelial tissue and connective tissue. They're non-myelinated, small diameter group C fibers. Temperature, pain, light touch are usually non-encapsulated or free nerve endings. Like this one, right? That's a free nerve ending. That's a thermoreceptor. Your body will, these sensors will sense cold in this range here. They're found in the dermis and actually in the papillary layer. And if you remember the dermis, those are those little finger-like projections, which on top here would be epidermis. And the bottom here would be dermis. Well, in fact, the free nerve endings are kind of located here in those papillae, along with the nerves in the blood vessels. So those are going to sense temperature changes on the surface of the skin, in fact. Again, for cold, about 10 to 40 degrees Celsius, and for heat or hot, 32 to 48. Okay, those receptors are located further in the reticular layer of the dermis. Now, both of those will indicate cold versus hot. If we go outside this range, so under 10 degrees Celsius or above 48 degrees, then the body's going to perceive those temperature ranges and those outside of those ranges as pain. And that is where the nociceptors will receive that pain, in fact. And this, in fact, is why when it's really cold, the body perceives coldness as pain, or if it's really, really hot, then you can, in the same regard, the body perceives that as pain as well. Nociceptors, excuse me, are free nerve endings as well. And those are pain receptors, right? Again, we just talked about extreme temperature changes, extreme pressure, like a pinch, or chemicals being released. Now, chemicals get released, for example, when we damage tissue. So when there's tissue damaging effects, the chemicals that release from those cells actually get received by the free nerve endings, and you perceive it as pain. So there's one thing that happens when tissue gets damaged. And so you see that. And the other one is, of course, the inflammation response. When you see swelling, it pushes against some of these nerve endings, and that causes pain as well. The tactile or Merkel disc, right, that we found in the skin is a light touch receptor. We have hair follicle receptors, which are free nerve endings that actually wrap around the hair follicle. And these are light touch receptors. When you bend the hair, we'll receive that change, right? Now, in fact, a mosquito is so light that the other nerve endings cannot feel the touch, per se, or we're not as sensitive to that as other heavier objects. But when the mosquito moves the hair, that's when we actually feel the mosquito, in fact, on our skin. It's from the hair follicle. Okay, so this table here, table 13.1, shows you the different types of receptors there, and we just really talked about these two at the bottom, right? Free nerve endings, hair follicle receptors, and some examples of which. And you see the different structures there, right? Different kinds of receptors. Okay, so these are the encapsulated ones. Those are the free ones, non-encapsulated. These are the encapsulated ones. Mostly mechanoreceptors. So when we talk about encapsulated, there's actually a connective tissue that surrounds the edge or the end of the receptor. And these respond to changes in touch, pressure, again, physical movement, right? Tactile or Messenger's corpuscles. Are the receptors involved in discriminative touch? Mostly in sensitive, hairless areas like the fingertips, where you have going to be, where you're going to have Messengers or tactile corpuscles. And laminar or Pacini corpuscles, deep pressure and vibration, deep dermis. So light touch, deeper touch, vibration, pressure. Again, both of which are mechanoreceptors. Bulbous corpuscles or Ruffini corpuscles, deep and continuous pressure. Muscle spindles are actually spindle-shaped proprioceptors, and they are wrapped around the muscle to respond to stretch. Tendon organs, they're actually proprioceptors that are in the tendon that detect stretch. And again, we're just kind of specifying where they're at. And joint kinesthetic receptors or proprioceptors that are found at the joint. And And this table 13.1 just kind of summarizes each one of those. There's the tactile, the Pacini corpuscle, the Ruffini corpuscle, muscle spindle, tendon organ, and joint kinesthetic receptor. Name, where they're found, right, and their function. Okay, so we're going to kind of switch this over and talk about how we process sensation, right? And again, this is really engineered because of this, right? Survival. And when we look at survival, we look at how this works because, again, every living organism is related to one another. This comes from an old part of our engineering, if you will, in the body. In the fact that we need to perceive information from the outside world so that we can survive, and not only that, but including the inside of the body. So we then can survive so that we can live, so that way we can satisfy the goal of every living organism, which is to reproduce, right? That's if you survive, you have a better chance of reproduction. Okay. So again, sensation is the awareness, right, of the internal and external environment, right? Perception, the word perception, is the awareness, conscious interpretation of those stimuli. Okay? So let's look at a type of reception that we have inside the body that we call the somatosensory system, right? These are going to be all of those types of receptors, like the external ones, the internal ones, and the ones that locate our body in space and time, are all fall into the somatosensory system, right? Okay, so in a sensory system like this, right, somatosensory system, you've got the receptors, right? You've got how this stimulus or this information is going to be processed up the way into the CNS, and then how it's going to be perceived, right? How it's going to be processed, right? And we go back to the cerebral cortex, right, of the CNS. So we go back to that, right? Because that's really where this information's going, right? It's going to go through the brain, obviously in the circuit level, but it's going to get processed in the cerebral cortex, right? So again, we're just looking at kind of a repetitive theme where you see reception or sensory, interpretation, processing, right? But you don't see the output yet. We're just looking at how it's going in, how this is getting processed, how it's getting sent from the sensory side or the sensory receptors up into the CNS. And so this figure does a really good job of kind of putting this together because it's giving you 3 examples on the receptor level. This is the receptor level at the bottom here. This dark brown square is the receptor level. And it's giving you reception from 3 places, right? One being the skin, one being the muscle, and one being in the joint. And 3 different types of receptors, right? There's the free nerve ending at the skin level, and you'll see it right at the dermis, where you see the papillary. That's where the black lines really begin, right there in that circle. You see the muscle spindles, those black kind of looks like circles, and in fact they're surrounding muscle fibers right there. And then you see those receptors there in the joint kinesthetic receptor right at the joint level, receiving information there as well. And you can see that all three, in fact, these black lines represent the neuron, in fact, receiving information and sending that information, localizing it, and then sending it up into the cerebral cortex through the second part, which is the circuit level, which is in yellow there. And the circuit level is actually processing it, localizing it, and then divvying it up or sending it where it needs to go, right? So you can see it kind of all three of them become one right here, but in fact, it's not how all the neurons are kind of spliced together. What that really means is that they're actually bundled together. And we're bundled together in what we call a nerve, right? And that nerve actually goes into the spinal cord, and you see this as these pathways or these tracks in the spinal cord. These areas where all these neurons are bundled together, in fact. We don't consider it a nerve yet, or nerve in the spinal cord, we consider that a track, right? And that's what you see there. And if you notice, there's a certain part of the brain then after the spinal cord where certain places stop, right? Certain neurons stop, like right here, whereas other neurons will stop or end at certain other places of the brain, and you'll see other neurons end, in fact, at other places of the brain. And then you start to see other neurons interconnected, right? You see that one there. You see these three here, right? You see this one here. And those are different neurons, right? Again, neurons connect to other neurons, right? So that's what we see this little on the surface. And in fact, goes like this, right? And then it has this here kind of thing, where that these are the dendrites. So that would be the cell body here. This would be the axon. This would be the end bulb or the end plate. Does that make sense when you see the, so we're going from here all the way up here to where you see these little here and there, right? The little dot represents, where you see a dot represents the cell body of another neuron, right? So we're all again, several neurons are interconnected there. But then finally you see the perception level. So we're sending finally and splitting up depending on what kind of stimulus, where it goes in the somatosensory cortex or the cerebral cortex. And you see that there, right? You know, the somatosensory cortex is going to be found there, right? And it just depends on what kind of reception there is, what place of the cerebral cortex that's going, right? Depending if it's coming from what part of the body and what sensation that's going to be. Okay. So at the receptor level, in fact, the neuron generates a signal, right? The stimulus has to excite the receptor. A graded potential is created, and then an action potential is created, and that has to reach all the way to the CNS. Has to go from the nerve, wherever that's going to be, up into the spinal cord. We call that process transduction, right? The energy of the stimulus causes a graded potential. The graded potential has to reach a threshold, and that threshold generates an action potential. Very simply, right? Same type of process that would occur that we saw in previous chapters, right? Again, that's adaptation, right? This is the sense of, for example, that you can feel vibration and after a while it goes away, or pain for a while and then pain begins to degrade or stop. Well, it's because Some of our receptors have adaptation, so they don't perceive the stimulus anymore. And because of that, we can resist, for example, pain, pressure, touch for a while. And so that'll stop. Okay, so when we process at the circuit level, right? There's different types of neurons, right, in the processing of the circuit level, right? Depends on where you're at in this process. We then give a name for each one, right? So again, we're going from sensory to then the cortex, right? And so if you're a first order sensory neuron, you're taking impulses from the receptor, right? To second order neurons, right? So you're the first neuron that's receiving the sensory reception. At your end or your beginning, you're going to have your receptors there, right? You're going to have those endings, free nerve endings, whatever they are. You're going to conduct that information. You're going to give that information to the second order neuron via neurotransmitters. And the second order neuron is a go-between because it's going to transmit the impulse to the third order. And the third order conducts impulses from the thalamus, because the thalamus is our kind of relay station, to the somatosensory cortex or the cerebral cortex. So most first order neurons that's PNS, right? This would be considered CNS in the second and CNS in the third. That's what we're seeing there, basically. Okay, let's talk about pain, because pain is really important in the human body because everybody perceives pain, knows about pain. It's actually engineered to have an awareness of pain, right? So pain in this regard, right, warns us of tissue damage so we can protect that tissue, protect that organ system, whatever the case may be, from further damage, right? That's really what pain's function is in the body, right? So extreme pressure, temperature, chemicals like this, right? Those can all be considered and perceived as pain, right? Some of those we release in the body. Sometimes when we damage tissue, those get released. Some of them are part of the immune response, right? The nerve fibers, or the neurons, right, release these neurotransmitters here, which we haven't really talked about neurotransmitters other than acetylcholine, right? And we won't discuss pretty much any other ones but these two, right? Substance P and glutamate are the neurotransmitters that pain impulses or pain fiber neurons will release, right? And releasing these to other neurons will then cause the perception of pain, right? Okay, so let's talk about tolerance, right? We all perceive pain at the same intensity and you know, the same type of process, right? But the tolerance will vary depending on the person, right? And this is depending on how many chemicals that we release and how many those releasing chemicals block the reception. This also depends on the adaptation. Some of our bodies, depending on the person, can adapt to the pain and not be sensitive to pain. So there's a big difference between pain threshold for some people. When we talk about visceral pain, this is where we're getting pain from the visceral organs, right? This is the burning sensation you would feel inside of your body, gnawing pain, aching pain. Many different things, but muscle, for example, spasming, chemicals getting released from inflammation, blockages of blood flow, ischemia, tissue getting stretched would be reception that you would perceive as visceral pain. Referred pain is commonly used in medical science because we can use this as a tool to actually identify where a particular tissue damage would be located, or malfunction or abnormality. And we see this as a body region, right? Pain from a body region perceiving as coming from a different body region. And so this figure here, 13.3, talks about looks at the referred pain on a map, right? And so these areas, for example, and you can see this one, this is a textbook one here, the heart, right? Where you see pain on the left part of the chest, left part of the inside of the arm, left part of the neck and the head. And that's a textbook sign of a stroke or heart attack, right? But then you can see some other ones, and I'll just show you this other one here, right in this area, which is indicative of kidneys, even though that pain can actually radiate down the thighs, inner and outer. Most of the abdomen and the pelvic area can still be perceived as, as pain in that general area, but it's actually probably radiating from or originating from the kidneys. Right, so we see these many different, parts that we consider referred pain. Look at this one right here, liver. Right there in the chest, and then over on the right side of the shoulder. Okay. Let's look at that, right? How we transmit. Talk about nerves, right? All right, so a nerve is a cord-like organ of the PNS, right? Again, this is a bundle of many different axons. They can be myelinated or non-myelinated. We've got connective tissue covering them, right? And again, we've got spinal nerves and we have cranial nerves, right? Here are the connective tissues that cover a nerve, right? So they go from inferior to then superficial in that order, right? And again, this is talking about the connective tissues. And then we still use the same word in science there to describe the location, but then we change, as in muscle, but we change the last word there, neurium, instead of mycin, right? But still talks about then the coverings, right? So you see a nerve here, and in fact it's a cross-cut of a nerve. The interior part, the middle part, is the blood vessels, so you see blood vessels there. And then you can see that we also use the word fascicles to talk about the bundling of these nerves, and each one of these dots represents a nerve. And we can't indicate yet from this picture which ones are sensory nerves and which ones are motor, but Nonetheless, we see the nerves there. I'm sorry, the neurons. That's what I meant to say. Each one of those are neurons. Right? So we see the bundling here, if you will. The individual bundling of a neuron is an endoneurium. And then we have the perineurium that surrounds several bundles of neurons. That would be one bundle would be a fascicle. And then this whole thing that surrounds connective tissue that surrounds the entire nerve is called the epineurium, right? And that's what you see in the book, and that's what you also see in the lab book, right? These 3 coverings, right? And then you see one neuron and its axon pulled, right? Pulled out. So some of these bundles can be, for example, this bundle right here could be sensory neurons. And this bundle here could be motor. I'm not saying it is, it's just could be there. And that's how we bundle and separate the sensory and motor all bundled in one nerve. Because this is all PNS, right? This does not describe the CNS. This is PNS nerve. Again, most nerves are mixed, right? We've got the afferent and efferent, the input and the output, right? Now, as I mentioned, most of them are mixed, right? Sensory and motor. But some of them are just located or isolated just for the sensory information, and some of them are just for the motor information. And we don't need to know which ones are which, but we just need to know that most of them are mixed, and some of them are sensory and some of them are motor. We've identified this word already, just to mention it again, right? Ganglia, right? Those are cell bodies that are associated with nerves in the PNS. Well, the cell bodies are kind of bundled together as well, because when we talk about a nerve, it's mostly the axon, right? But the cell bodies are also bundled together, right? So, for example, in the sensory neurons, right? The cell bodies are the ganglia there. We call those the dorsal root ganglia. And sometimes we can identify them depending on if they're autonomic too, right? Those are the autonomic ganglia. And this one here, let's talk about the regeneration, right? How we actually can do this, right? So when neurons become complete, right, or matured, right, they also become amitotic, incapable of undergoing mitosis. So they lack the capability of dividing if they've gotten damaged, or multiplying, if you will. And so that's one of the reasons why neurons cannot be regenerated. But in the PNS, we have a special capability of regeneration, but only a certain part of the actual neuron, right? So if the cell body doesn't get damaged, but the axon gets damaged, the axon can regenerate, right? So just in the PNS, not in the CNS. So only PNS axons can regenerate, and that's only if the cell body isn't damaged. If the cell body is damaged, then the cell body and the entire neuron will then not be able to regenerate. So why doesn't this happen in the CNS? Well, it has to do with the oligodendrocytes. The oligodendrocytes actually have this protein that they secrete that prevents this regeneration. The astrocytes actually have the capability of forming scar tissue. And this is what can occur in a repair, if you will, or because it's not really repair, I wouldn't use that word repair, but this is what happens when a neuron gets damaged in the CNS. Let me put it that way. So the PNS is what we want to look at when we talk about regeneration, right? And it also depends on the level, the severity of the damage. So there can be cases when the damage is so severe that regeneration will not occur in the axon no matter what, right? So here's what occurs, right? The place that is damaged, let's say the axon that's damaged, right? We're going to form these pieces of the axon, right? So the myelin sheath that's damaged, the axon itself, the plasma membrane, organelles that are broken apart, disintegrate, right? The cell has the capability, the body has the capability of kind of breaking this stuff down, which it happens, and we call that Wallerian degeneration, right? And we start to kind of basically clean up or edit all of the damaged part of the axon, right? And that's the first step, right? First process. Macrophages actually eat away at all of the damaged parts, and they'll begin to degrade that because that's what the macrophages are doing. The Schwann cells are there, and the Schwann cells will divide or replicate. The axon will begin to actually propagate or grow, right? We call this a regeneration tube, and we see this step when we see the axon in the plasma membrane beginning to be reformed. And then we see the regeneration and the new myelin sheath forms over that area. Right? So here's step one. Here's where we're at, right? We see that part of the axon. And say that part of the axon got damaged. Let's say, I don't know, let's say heat was applied. Really hot temperature was applied here. Caused that to break open just like it did there. Let's say that that's the damage, the source of damage, right? Enough heat was applied, you burnt the axon, this is what occurs, right? So what's occurring here is Wallerian degeneration, whereas these pieces of axon, these pieces of myelin begin to get degenerated, right? So they begin to be broken apart. Macrophages will come in and start to physically eat away or undergo phagocytosis. All of the old broken up pieces. This is what you see here. And then instead you start to see these filaments forming. We see this, we call this whole thing right here a regeneration tube, right? Where we see plasma membrane forming these kind of little long strands, these filaments, which will actually meld together and become axon, right? And you'll notice the blue cells are Schwann cells. Those are Schwann cells there. And they have to undergo division, so they actually do undergo mitosis and form extra myelin there, wrap around the axon, and this is where repair will occur. So we see that there.